This is Team 18, consisting of Jeremiah, Maddie, Ethan, and Nathan. And today, we're going to be presenting immune response.
Ethan is a cell in the body. Sometimes, when cells come in contact with dangerous things like viruses, such as this floor patch right here that's exposed, dangerous things happen.
Help, I've fallen and I can't get up.
Right here you see the floor patch and a cutout of a carpet that fits just in place, like an antibody will fit in right with the virus or bacteria that it's trying to destroy. And now, the bacteria or virus is no danger to our cell.
Immune response is basically how your body reacts to an infection, such as viruses or bacteria. Your body's main response to infectious diseases is phagocytes. Phagocytes' job is to eat and dispose of any material that your body doesn't want inside of it. Occasionally, phagocytes get overwhelmed with the amount of infectious diseases inside you, and they need help. When phagocytes need help, antibodies come by to help. Antibodies' job is to stop and hold up infectious diseases so they can't harm your body anymore, and so they can't avoid the phagocytes. How antibodies stop infectious diseases is an interesting process. Antibodies attach onto what's known as an antigen on the outside of the disease. Whether it's a virus or a bacteria, they have antigens on their cell membrane that the antibodies can attach onto. Antibodies also have chemicals on the outside of their cell membrane that allow them to attach onto the antigens. Once the antibodies have swarmed around the infectious diseases and they've attached onto the antigens, the infectious diseases can no longer harm your body and they have more trouble moving around. Another unique feature of antibodies is their ability to attach onto phagocytes. Because the two work together, it's easier if they're in the same place. After the antibody attaches onto the antigen, the phagocyte's right there to eat it. Different diseases in different situations call for different types of antibodies. There are 5 main types of antibodies. All antibodies are known as immunoglobulins. There are 5 types: A, D, E, G, and M. G is the most common immunoglobulin and is shaped like a Y. The unique Y shape of immunoglobulin G allows it to create chains of immunoglobulin Gs and infectious diseases together, preventing more of them from moving around.
So I'm going to be talking about viruses and more specifically how they interact with the cell membrane to then go on and infect the cell. So here we have a general idea of how the virus comes in, then attaches to the cell membrane. But in order to understand how this binding really happens, you need to understand just a little bit about proteins. So usually in a protein you'll have the part that makes it function is the binding site or the active site. And this a lot of times takes place in a hydrophobic region, which will usually be in a pocketed region within the protein. So the way that this interacts with what we're talking about today is there are several amino acids that stick out into the hydrophobic region that do have charges on them that will allow it to attach to specific molecules that it needs to. So in this case, the protein attached to the virus is hemagglutinin. It's usually called HA. And it is looking for sialic acid, which is attached to cell membrane proteins. So if we look at the structure of sialic acid here, we see a lot of oxygens and hydrogens along the outside of the molecule. So although the hydrophobic pocket doesn't usually accept many charged particles in there, it's really just gonna keep out everything until it finds specifically what it's looking for. So now that we understand a little bit how those function with each other, where the antibodies come into this is the antibody will be trying to imitate the sialic acid. For hemagglutinin, the main antibody that we look at in type A flu is going to be called HA7. So in this next image, you can see a little bit better the structure of the protein. So what it's going to do is, like I said, try to imitate the sialic acid. So the antibody can bind onto the virus, and then the antibodies signal to the white blood cells to come and destroy them. In the next section, more on bacteria, you'll get a little bit more of the mechanisms for antibodies and other kinds of immune responses.
Bacteria are single-celled microorganisms that are present in and around our bodies, most of which are harmless or even beneficial. However, certain disease-causing bacteria can cause serious damage to body cells and function when they infect a person. Bacteria are much smaller than our own cells, but much larger than viruses, and consequently have more complex and diverse ways in how they cause infections. Some bacteria attack cells and tissues directly. Some reproduce and overcrowd body cells, making them unable to function properly, while others release toxins into our bloodstream when they reach critical population densities. Bacteria have even evolved a communication method to identify these critical population densities, known as quorum sensing. Quorum sensing is a mechanism of microbial signaling where microorganisms, like bacteria, synchronize their behavior depending on their population densities. Bacteria release chemicals as metabolic waste and also through active signaling processes. They are also able to reuptake and recognize these signals from other microbes. As the bacteria population grows, the concentration of these signaling molecules in the immediate vicinity increases, and once the specific concentration threshold is reached, the bacterial colony can respond in a synchronized way, such as releasing toxins or attacking nearby cells. There are three main classes of signaling molecules bacteria use for quorum sensing: oligopeptides, N-acyl homoserine lactone, or AHLs, and autoinducer 2, or AI2s. In the case of oligopeptide signaling, a pre-protein is generated that is subsequently processed into the active signaling peptide, which is exported from the cell. The length of the pre-protein chain varies in different systems. In some cases, the peptide might be active, whereas in other systems, the peptide is modified to be active. Oligopeptides are the largest of the signaling molecule classes and are transported out of the microbe through active transporter proteins. N-acyl homoserine lactone, or AHLs, are common in gram-negative bacteria. Specialized proteins, including the ACL carrier proteins and AHL synthase, regulate the production of AHL. AHL signaling molecules have very similar structures as a result, which enables mutations to these signaling molecules in a population of bacteria to be easily recognized and responded to. The structure for AHLs include a lactone ring, an amide group, an acyl chain, frequently containing an oxo or hydroxyl groups at the 3' position, as well as levels of unsaturation. Alterations to these side chains allows for a diverse number of signaling molecules, each usually characteristic of a specific bacteria strain. It is mutations to these chains that allow for rapid evolution of bacterial communications for strains which use AHL signaling molecules. One example of this is the species P. aeruginosa, a multi-drug resistant bacteria that usually infects a host with compromised immune systems. It can cause serious infections in people who have cystic fibrosis, as well as cause pneumonia and skin infections after severe burns. These structures are two known AHL signaling molecules for P. aeruginosa, in which we can clearly see the functional groups indicative of this class of signals. Its rapid evolution of quorum sensing signaling molecules enables it to evolve immunity to antibiotics over a very short period of time, making it very difficult infection to treat.
One of the most common and important signaling molecules for bacteria is AI2. It is sometimes referred to as the universal autoinducer, as it is found in many gram-positive and negative bacteria. AI2 is unique as well, as it is the name for not just one molecule, but many derivatives of the compound DPD. On this slide, you can see the relation between the many DPD derivatives. Here on this slide, you can see the biological synthesis pathway that bacteria use to create DPD. It is important to note the mechanism of this, as one of the ways to inhibit the production of DPD is to disrupt this mechanism. Although there are many possible ways to inhibit the production of DPD, we will focus on the removing of LuxS, which disrupts the production of DPD. This slide shows two possible mechanisms on how a brominated furanon molecule can be used to reduce the functionality of the LuxS molecule. Both mechanisms show how a nucleophile from the LuxS molecule, which is represented by the squiggle line, adds onto a nucleophilic region caused by the deficiency of electrons created by the bromine. This adds a large molecule onto the LuxS molecule, severely reducing its ability to create DPD. And once DPD cannot be produced, the bacteria lose their ability to communicate with each other, and are therefore much less dangerous to the person they are infecting. These mechanisms for intermicrobe communication make bacteria a serious danger to our body's health. Like viruses, they reproduce until they overpower our own cells' abilities to function, the primary difference being that bacteria replicate outside of cells, while viruses replicate inside of them. Our immune system can attack bacteria in a very similar way to viruses as a result, initially having macrophages consume them, and sending in helper cells and antibodies when the infection overpowers the macrophages. Opsonization is the most common method used by antibodies to destroy bacterial pathogens. It involves the binding of an antibody to an epitope of an antigen. After the antibody binds to the membrane, phagocytes are attracted to the pathogen. The Fab portion of the antibody binds to the antigen, whereas the Fc portion of the antibody binds to the Fc receptor on the phagocyte, facilitating phagocytosis. Here we see a close-up of the binding sites on an antibody. The Fc region on antibodies is the area that allows them to interact with the receptors on the white blood cells to fight infections. The Fc stands for fragment crystallization region and is constant throughout all antibodies, as it only has to interact with our own cells. The Fab portion is variable on the antibodies and determine what is the specific invader they can target. Here on this slide, you can see another in-detail image on how the antibodies bind to phagocytes with the Fc regions. The gray and white chains are the Fc region on the antibody, while the magenta, orange, and cyan sections are the Fc receptor on the phagocyte. In the two close-up sections, you can see the Fc region on the antibody, shown as sticks with carbon in yellow and red as oxygen, and how it interacts through intermolecular forces with the cyan Fc receptor on the phagocyte. This occurs through the attraction of only certain molecules in certain areas, like the hydrophobic region, shown as the area with the red dotted line around it, where the nonpolar carbon chain LEU-235 can be inserted.